Today's topic is a very good topic and it relates to the upcoming area. So, dermatology segment is the upcoming area in the field of Medicare, right? And it is growing at the rate of 20% per annum. So, wide scope. Anyone who knows how many product a man uses in a day, a male uses in a day, any idea? We start uh, our morning with the hand wash and toothpaste, right? Everyone is using, whether it is a male or female. And male uses 9 plus cosmetics product in a day, and female uses 15 plus product in a day. So it is a huge market, domestic as well as globally. So this was, if you go 30 to 40 years back, there was no trend of using the cosmetics. There were hardly one, two brands like Lakme and Pond's cream was there, right? Not too much products. And there was a local face cream, that is a snow cream was there, Afghan snow cream. And all were conventional formulations, all were conventional. So now the time has changed, right? There is a modernization in the manufacturing as well as in the use of the product, right? The modernization is with the lipid and lipid nanotechnology, that is a liposomes. So still people are not much aware of the liposomal formulations. So hardly 1 out of 20 formulations there is a one liposomal formulation available into the market. And rest are all are conventional. The reasons may be due to high cost production cost, right? High raw material cost and less awareness among the consumers about the liposomal products, because nowadays consumers expectations has also been increased. If someone is visiting to dermatologist, his first question is, doctor, in how much time I will be okay, my skin will be okay, right? So this is the first question of the consumer to the dermatologist. So new technologies has a very are the products which are manufactured with the by using the new technologies, they are safe and efficacious. And CDSO, the regulation making authorities, they are focusing only two things. One is safety and another is efficacy, whether it is a cosmetic or it is a drug. But unfortunate part in our country is that we are having only 15 to 20 pharmaceutical formulations. Those are liposomal one, right? Or there is a use of lipid nanoparticles. So lipids are just you can say it is a fatty material and non-polar one, right? And we use the lipids to create the nanoparticles, lipid-based spheres, which facilitates the delivery of drug and cosmetics. So these are the basically carriers of the actives or pharmaceuticals ingredients, right? It's a one of the novel drug delivery system. So these are some properties of nanotechnology. So lipid nanoparticles are widely used to deliver therapeutic agents, protects mRNA from degradation in the body and facilitates their transport into cell. So biggest problem uh, in testing of the formulations, particularly dermatology formulations in our country, is that we don't have any protocol to test the dissolution rate. That is a subcutaneous penetration or dermis penetration rate of the material. There is no set system or no equipment available in the with the government institutions as well as with the private institutions. Those can test the dissolution rate of the uh, external formulations used for external preparations. So that could be the reason. There are some products available into market. They are having misleading claims. They may not be adding the liposome somal material. They must be using something like lecithin, soya lecithin, etc., and claiming it is a liposomal one. So I was talking to Mr. Kedia, Arun Kedia ji, that how much consumption of the liposome is there in domestic liposomal consumption is there. So he was just telling me that 94% they are exporting and 6% consumption, their consumption is into the domestic market. So globally, the size of the segment is about and if we take the data of 2025, it is a 5 billion US dollar nearly, and it is expected to increase up to 12 USD US million dollar up to 2032. So this is the segment is growing at the rate of 6%, whereas the pharmaceutical segment is growing at the rate of 4%. So surface of lipid nanoparticles can be modified with the specific molecules to target particular cells or tissues, reducing of targets, side effects, and enhance efficacy. So this is like some hair formulations for the growth of the hair, or anti-hair fall formulations, or for growth of the hairs. This salt sphere technology, that is a silicium ion. The active is being carried by the silicium ion, and it facilitates the penetration of the active into the scalp. So, in manufacture of such formulation, one has to take care that the size of the your lipid nanoparticle matters, size and shape, right? Liposome matters, because the size of the scalp pore is about 40 micron in size, so it should be less than that, whereas that of the skin skin pore is less than 28 micron. So see, which is the biggest organ of our body? It's the skin. Skin has a length of our total body skin length is 8.5 meter, right? And we are using about 20% product for the our body, whereas the 80% is used for the on the face only, right? Maximum product we use on the face, cosmeceuticals. So then this lipid nanoparticles can be used to reduce the side effects and to enhance the efficacy and as well as of the safety of the product. The examples are your sulfurous caffeine, benzoyl peroxide, as well as of the salicylic acid. So benzoyl peroxide loaded molecules are available by one of the company, that is Salvona. Salvona is a trendsetter in salt sphere technology, that is a salt sphere caffeine, right? It is a caffeine is a hair stimulator, right? And then benzyl peroxide is used for the acne, so this is available into the matrix embedded form. Benzyl peroxide is known for its disqualities as dryness of the skin, right, as well as it causes some sort of irritation also. So whereas this is being reduced by the use of salt sphere technology. So same is the case with the salicylic acid. Salicylic acid is being used as exfoliant for the in the skin formulations, and the irritation of salicylic acid is being reduced by adopting this technology. So these are the three types of lipid nanoparticles are there. which we are using into the manufacture of the cosmeceuticals as well as into the dermaceuticals. This solid is being used into the oral dosage forms, right? But this is again being used by most of the nutraceutical manufacturers, right? But in pharmaceutical, there are very few formulations available into the market. Then our main topic is liposome, because in the skin formulations, dermatology or for hair care formulations, personal hygiene care products, we are using only liposomes. So this is how liposomes work. The size as well as shape of the liposomes matter. The shape of the liposomes for incorporating into the skin formulations, that should be multilamellar shape, and size should be less than 28 micron. So this slide shows how multilamellar vesicle penetrates through the skin, through the dermis. So the uh, structure of our skin is as phospholipid, bilayer, and phosphatidylcholine. So this is one of the product which we have developed as a liposomal. This is a metformin serum which we have introduced for the first time, not only in the country but globally for the first time. Metformin, this we have developed into in our own lab. This is a liposomal formulation. Metformin is a otherwise oral antidiabetic drug. So the data is available. This metformin solution topically 10 to 30% of its solution is effective for the treatment of chloasma. Chloasma is the hyperpigmentation which occurs during the pregnancy stage, and then acne inversa, and then in the deep-seated hyperpigmentation. So this is one of the model which shows how the liposomes give hydration to the skin, right? For the treatment of atopic dermatitis as well as for the psoriasis, right? There is a need of some sort of hydration in the on the skin, right? So this is liposomes. This holds the moisturization into the layers of the skin. So this all you know what is liposome encapsulation, that I have shown in my earlier slide also. So this I was talking about the benzoyl peroxide, saltsphere benzoyl peroxide. So in fact, these are the loaded embedded matrix are available in the market, loaded with benzoyl peroxide, and 25% of benzoyl peroxide is being loaded into the matrix, in the matrix forms, right? Similarly, niacinamide liposomal is available in the market, right? And this has a one additive quality that it does not causes any irritation and does not causes any skin any dryness to the skin and irritation to the skin, and moreover it has a sustainable action for 24 hours. So the consumer gets relief from the acne and from the acne outburst within a period of 5 to 6 days. So these are the stability challenges for the liposomal formulations, right? The little bit unfortunate part, I am not condemning anyone. The production chemists are facing these, but they do not know how to overcome these challenges, right? It is difficult to manufacture the liposomal formulation and difficult to stabilize it, right? What the manufacturing chemist does, whenever they are making some cream or gel, they add the liposomal product at a high shear speed, so the liposomes gets broken and the purpose get defeated, right? Then they do not control the temperature, whereas this is to be liposomal ingredient is to be added at less than 40-degree temperature, right? The what the production chemist does, they add the liposomal material during the processing of the batch, wherever it is required to cool down the formulation and then to add the liposomal material. The another thing is to add the antioxidants to prevent the oxidation, right? Vitamin E is there, vitamin C is there, then BHT is there, that can be added to check the oxidation. And then pH, setting up of the pH is most important for the liposomal formulation. So once it is on the acidic side or it is on the neutral side, the liposomes will not work, right? They will become unstable. So the best pH is range is between 5 to 6, 5 to 6. So about the temperature, I have already told you, because this liposomes are thermolabile in nature, of course. Then buffer species, they are sensitive to buffer species also. So one more thing is to stabilize, how we can stabilize uh, these liposomal formulations. So that is the addition of cholesterol into the formulation, then surface modification by coating with the polymers or surfactants, right? Then lyophilization, that is addition of cryoprotectants like propylene glycol, ethylene glycol, sorbitol, etc. Or you can add citric acid also. Then oxidant, I have already told you. Then the important role is the humectants. There is a fixed percentage of humectants is there that is to be added into the dermatology formulation, that is a cosmeceutical formulations, which stabilize the liposomal formulation. So what are the advances in lipid nanotechnology? So this is not the case that we are using the lipids right now, but the form has been changed. We were using the lipids, or doctors were recommending the lipids 30, 40 years back, also 50 years. Whenever someone, patient was taking some antifungal, they used to recommend take this drug uh, with milk, right? Or take the milk or butter and other fatty materials in your diet, right? Because the most of the antifungal drugs are lipophilic in nature, so they become the food. Your food itself was a carrier. Foodstuff was itself a carrier of the drug molecules. So few examples of this liposomal products in the pharmaceuticals are like cisplatin, cyclosporin, and doxorubicin. So these are the advanced therapeutic strategies. You see, nowadays, you must have seen some cancer patient. So most of the cancer patients die due to severe pain. So nowadays, the trend is shifting towards the dermatological preparations. That is either your intradermal patches like fentanyl patches are there, right, to reduce the pain. And then your one of the buprenorphine. That is of course a narcotic drug, but it is a very good drug for as a pain reliever. So they gives a continuous concentration of the buprenorphine or fentanyl, right, and they maintain the peak plasma level, and there is a no pain to the cancer patient. So similarly for chain smoker, nicotine patches are there. So this is one of the most convenient route, right? And in this, the you can say the precision of the dose is there by this route. And third is it does not goes to the hepatic. Hepatic does not passes to the liver. So this is the regulatory status of the liposomal drugs. Once you develop a liposomal drug, you cannot market it, right, directly from getting the license from this SLA, State Licensing Authority, because you have to, because that is covered under the definition of new drug under the provision of Rule 122E. So you have to take the permission of DCGI by submitting the data of safety and efficacy. Safety and efficacy data. So this is a somewhat cumbersome procedure, right? That could be the reason that there are 15 to 20 formulations, pharmaceutical formulations are available into the market. So this slide shows a continuous research is going on in the Punjab University Institute of Pharmaceutical Sciences. Professor Katare is actively engaged, Professor Bhoop, who is working on QBD also, and then Professor Indupal Kaur is there, and 6 to 7 products are already into the market, which has been developed by the Punjab University and transferred the technology to the manufacturer. So here is the conclusion of using the LNPs, right? So I think the researcher should do more R&D to develop the new molecules and go to clinical phase trial 1, 2, 3, and then they can market the product, right? Since there is a lot of need to the nephro-oncology and other dreadful, the patients who are suffering with the other dreadful diseases. So thank you very much.
Thanks, Dr. Pardeep. So we can have a couple of questions, a question or two. So maybe Dr. Bachao and then Rajdeep. So we'll stop with these two questions.
Thank you, sir, for a very interesting talk. Initially, you mentioned there is no dissolution test for the topical formulations, but often we do use the Franz diffusion cells with silica membranes. And that is something quite acceptable by US FDA or European authorities as a QC test. What are your takes? Use of Franz diffusion cells as an in vitro tool to check the release profile of the transdermal formulations.
You're right, sir. But see, under regulatory provisions, we don't have this test for the dissolution rate. We can't test. We don't have any equipment. None of our lab has any equipment. Government lab, government approved labs, they don't have a protocol for that. No established protocol. Even you see the Indian pharmacopoeia, you won't find any test for external preparation for such a dissolution test. That is only for oral dosage forms.
Yeah, thank you for your talk. So the question, yeah. So my question was similar actually. That what is the scientific challenge? For example, we in academia can take up to address the question of coming up with a protocol for dissolution through the skin layer. What can be the model membranes we can start? And of course, it has to be clinically taken up. That is the first part. And the second part was, as you highlighted, that there is no standard pharmacopoeia protocol also. Then how the formulations that are marketed are tested and benchmarked? That is my the
Sir,
curiosity question. Yeah,
yeah,
thank you.
my answer to your first question is that there is a process is going on to introduce this dissolution test for the external formulations, right, like ointment and creams, because at this time we cannot evaluate how much percentage of this active is penetrating into the skin or into the scalp, right? And I hope soon it will be introduced and it will be a part of our pharmacopoeia, right? The another thing is that at present the labs are testing only the active ingredients incorporated into the your ointment and creams only. Okay.
Yeah, this let's have the last one then.
Hello, sir. Thank you for your talk. I had a question regarding, as you said, the liposomal technology. But we face a consistent challenge of entrapment efficiency in case of liposomes. And to maintain that entrapment efficiency in each batch, how you are addressing that question regarding the manufacturing aspect of liposome?
See, if I will talk over here, it will be leaking of some sort of skill to manufacture the how to safely manufacture the liposomal product, right? But I have given you the hint how to stabilize. That is, you have to take the take into consideration the control of temperature, right? Then the speed of the you do not need to use the emulsifier in that case, homogenizer, right? You have to use only the mixer only when you have to add the liposomal product. Then you have to add glycerin, a set percentage of glycerin, that you have to do your R&D. I have done it, but you need to do it. When you will add glycerin into the formulation, right, it will stabilize the liposomes, right?
Just a short one, but and we can have more questions in the tea break then. But maybe just one last one then.
So you said liposomes are sensitive to buffers. So people use PBS for hydration of the lipids. So what should be the concentration? Nobody is mentioning the concentration of the PBS for hydration of the lipids to prepare liposome.
See, I have already told you the use of glycerol plays a very good role in that case, right? But you have to maintain the pH also. For that, you need to use the some weak buffers, right? Weak buffers only to adjust the pH, not the strong buffers.
Thank you.
So I think
Thank you, sir.
with this, let us thank Dr. Pardeep Kumar